Hello, and welcome to our fourth lecture in this series on physiological psychology. Today we're going to talk about neurotransmitters. In particular, we're going to talk about the sequence of events that occur at the synapse, talk about neurotransmission and neurotransmitters, talk about neurotransmitters and their receptor types, talk about different neurotransmitter systems, and then have a brief introduction to psychoactive drugs, and talk a little bit about hormones. Let's start with the sequence of chemical events that occur at the synapse. The major sequence of events allowing communication between neurons across the synapse occurs when the neuron synthesizes chemicals that serve as neurotransmitters. These can be synthesized in the cell body, or they can be synthesized in the axon or packaged in vesicles. There are a variety of ways in which these will be synthesized. So an action potential then travels down the axon, reaching the terminal buttons and the synapse. Those released molecules diffuse across the cleft, attaching to receptors and alter the activity of the postsynaptic neuron. So when an action potential travels down the axon, it opens calcium channels. Those calcium ions then come into the cell. That then causes these synaptic vesicles to fuse with the cell membrane, which causes the release of those molecules to diffuse across the cleft, attach to the receptors, and alter the activity of the postsynaptic neuron. The neurotransmitter molecules separate from the receptors, and then the neurotransmitters may be taken back into the presynaptic neuron for recycling or diffuse away and be metabolized by enzymes. Some postsynaptic cells may actually send reverse messages to slow the release of further neurotransmitters by presynaptic cells, and we call this autoception. So to give you a quick summary of this, we have synthesis of smaller neurotransmitters such as acetylcholine here in the terminal button of the axon. Action potential causes calcium to enter, causing the release of that neurotransmitter. That neurotransmitter then binds to the receptor. They then separate back out from the receptors. We get either reuptake into the presynaptic neuron. We get the metabolites of neurotransmitters broken down here in the synaptic cleft by enzymes. The postsynaptic cell may release retrograde transmitters that further slow release from the presynaptic cell, and we get negative feedback sites that respond to retrograde transmitters or to the presynaptic cell's own transmitter to stop release of the neurotransmitter. And again, this is called autoreception. So there are neurotransmitter sites on this terminal button that cause the ceasing of that release of neurotransmitter. Again, another basic summary of this, we have neurotransmitters are synthesized from chemical building blocks called precursors. Those are either synthesized in the cell body or in the axon itself. Neurotransmitters are then stored in these vesicles. The action potential cause vesicles to fuse to the presynaptic membrane and release their contents into the synapse. These released neurotransmitters bind to the postsynaptic receptors, and then neurotransmission is terminated by reuptake, enzyme deactivation, or autoreception. There are different types of neurotransmitters we'll talk about. This is a simple summary of the most common ones. Acetylcholine is involved in memory and stimulates movement. It's a particularly important type of neurotransmitter. Serotonin is involved in the regulation of our mood, our sleep-wake patterns, and our appetite. GABA, which is gamma-aminobutyric acid, is involved in sleep and inhibits movement. Glutamate is involved in memory formation. Norepinephrine is involved in arousal and mood. Dopamine is involved in control of movement and our sensations of pleasure and reward, and endorphins are involved in pain relief. So we'll start and take each of these steps that I've talked about already and break them down into some specific details. So the first is the synthesis of neurotransmitters. This is how neurotransmitters are formed. They are formed by the combination of precursors. These precursors are, of course, brought to the neuron by the bloodstream. These are usually different kinds of proteins. They are taken up by the cell body and/or terminals. They often come from substances in the diet like tryptophan or tyrosine. And then enzymes are used to put these ingredients together. And, of course, that's the role of cellular DNA and RNA, is to put these together into proteins. So neurons synthesize neurotransmitters and other chemicals from substance provided by our diet. Acetylcholine is synthesized from choline, which is found in milk, eggs, and different kinds of nuts. Tryptophan serves as a precursor for serotonin. This is one of the reasons why there is this lore about tryptophan in Turkey as the reason why you get sleepy after Thanksgiving dinner. And that's actually a myth. the reason you get sleepy after Thanksgiving dinner is because you've eaten too many carbohydrates, and that's what's really made you sleepy, is your body's trying to process all of those calories. It has little to do with the minor amounts of tryptophan that serves as that precursor for serotonin. The catecholamines contain a catechol group and an amine group. So this is epinephrine, norepinephrine, and dopamine, and again, these are derived from a variety of proteins found in our diet. Those neurotransmitters are then stored in vesicles. These vesicles are used to concentrate the neurotransmitters into small packages and to protect them as they are transmitted down through the axon so they don't get metabolized or used in other processes. They're released through a process called exocytosis. This is where the vesicles fuse with the presynaptic membrane and release neurotransmitters into the synapse. The binding then is the attachment of the transmitter to its receptor at both the postsynaptic neuron and the autoreceptors in the presynaptic neuron. This exocytosis occurs by the action potential reaching the end of the axon, opening calcium ion channels. Those calcium ions then enter the terminal button, causing the vesicles to fuse with the membrane and causing exocytosis. Transmission across the synaptic cleft by a neurotransmitter takes less than 0.01 microseconds, so it's really instantaneous. Most individual neurons release at least 2 or more different kinds of neurotransmitters, and neurons may also respond to more types of neurotransmitters than they release. So any one neuron isn't going to respond to just one type of neurotransmitter. Now we'll talk about neurotransmitter systems where they are the dominant part of a particular system, but there are a lot of different receptor types on neurons, and so that's something to keep in mind. So that's what we move to next, is to talk about different varieties of receptors and neurotransmitter type of receptors. The first of these that we are going to talk about are called ionotropic receptors, and that's because they act instantaneously and open ion channels. So you can see in this figure we have a closed ion channel. We have an ionotropic receptor and a neurotransmitter. This neurotransmitter binds, opens that this ion channel, causing an instantaneous change in the polarity of the neuron. We also have metabotropic neurons, which are very different. They involve changes in the metabolic properties of the neuron and the release of G proteins on the interior of a neuron. So the ionotropic receptors are the fastest responding type of neurotransmitter receptor. They cause direct reaction to the presence of the transmitter, so those ion channels open instantaneously. And they can cause a variety of actions or changes in the polarization of the neuron. So glutamate is our primary excitatory ionotropic neurotransmitter. Acetylcholine is another key ionotropic receptor which facilitates the opening of sodium channels. So both of these receptors will cause depolarization of the neuron membrane. GABA, or gamma-aminobutyric acid, is the primary inhibitory neurotransmitter. This works by opening chloride channels, allowing more negatively charged chloride ions to enter the cell, thereby hyperpolarizing the cell. Remember, when we're talking about a cellular membrane, we have to remember a few things, so it's worth doing a quick review. When we hyperpolarize a cell, it makes its charge more negative. So normally a cell is operating at about negative 70 millivolts. But when we get a hyperpolarizing of the cell, it pushes that cell even further negative. Depolarizing of a cell makes its charge less negative, moving it towards zero charge, and makes an action potential more likely. So depolarizing a cell, by allowing sodium ions in, makes an action potential more likely, and that's what glutamate and acetylcholine do. Whereas GABA is hyperpolarizing because it makes the charge more negative by allowing those chloride ions into the cell, and makes an action potential therefore less likely. Metabotropic receptors initiate a secondary signal or messenger inside the neuron by releasing what's called a G protein. So if you see here, we have a metabotropic receptor, and a neurotransmitter binds to that. We have this signal protein, which then bends and releases these proteins inside the cell. This takes further time and also has longer lasting results. So this initiates a secondary signal inside the neuron. So this occurs when neurotransmitters attached to a receptor initiate a sequence of slower and longer lasting metabolic reactions. Metabotropic synapses use many neurotransmitters such as dopamine, norepinephrine, serotonin, and sometimes glutamate and GABA. But these are far more complex interactions than the simple ionotropic reactions we talked about previously. So when neurotransmitters attach to a metabotropic receptor, it bends the receptor protein that goes through the membrane of the cell. This bending allows a portion of the protein inside the neuron to react with other molecules. Metabotropic events include such behaviors as taste, smell, and pain, whereas ionotropic events include things like vision and hearing. A lot of this has to do with how fast we need our system to react. Vision is a very rapid system, and so we need it to get shut on and off very quickly, whereas taste, smell, and pain are longer-lasting, more diffuse states. The reason why taste lingers, smells linger, and pain lingers is because these metabotropic events last for a longer period of time. So these are the sequence of events at a metabotropic synapse. We have a non-stimulated metabotropic receptor with a protein on the outside and on the inside. When that transmitter molecule attaches to the receptor, it bends, releasing that G protein, and that G protein then activates a secondary messenger such as cyclic AMP, which alters the metabolic pathway, turns on a gene in the nucleus, or opens or closes an ion channel. So that G protein released by the metabotropic receptor initiates a second signal inside the neuron. So this G protein activation is coupled to guanosine triphosphate, which is an energy-storing molecule. This increases the concentration of this secondary messenger, and that secondary messenger communicates to areas within the cell. This may open or close ion channels, alter production of activating proteins, or activate chromosomes. So there are a variety of mechanisms by which these G proteins might affect the behavior of a cell. We'll talk more about this when we start talking about things like epigenetic alterations of cellular DNA, which causes alteration in protein expression. So those are the different kinds of receptor types we're going to talk about. We'll talk a little bit about neuropeptides and neuromodulators, but they're kind of a special case. So how do we inactivate or terminate synaptic transmission? Well, there are three processes by which this may occur: metabolism, reuptake, and of course autoreception, and reuptake by glial cells, but this occurs for glutamate only. So neurotransmitters that are released into the synapse do not remain and are subject to either inactivation or reuptake. During reuptake, the presynaptic neuron takes up most of the neurotransmitter molecules intact and simply reuses them. It's the simplest way for this to occur is through recycling. Manufacturing of proteins is metabolically expensive, and so as a result, we want to try to save as much energy as possible, so the neurons simply reuse those proteins. Transporters are then special membrane proteins that facilitate that reuptake. There are also other properties by which this process is stopped, such as inactivation versus reuptake. So serotonin is primarily taken back up into the presynaptic terminal. This, by the way, is the target for most antidepressants, is blocking reuptake. So most antidepressants are what we call selective serotonin reuptake inhibitors. Acetylcholine is broken down by acetylcholine esterase into acetate and choline. This becomes important because acetylcholine esterase is a target for both beneficial drugs like drugs used to treat Alzheimer's disease, but also dangerous substances like nerve gas and pesticides. The difference is if they are sort of permanent or temporary blockades of acetylcholine esterase. Excess dopamine is converted into inactive chemicals by monoamine oxidase. Serotonin can be as well. So some drugs target monoamine oxidase to try to improve mood and etc. So we have CoMT, or enzymes that convert excess into inactive chemicals, as is monoamine oxidase. We also get negative feedback in the brain, which is accomplished in two ways. This is an important property by which cells learn to communicate with one another. Autoreceptors are receptors that detect the amount of transmitter released and inhibit further synthesis and release. We also get postsynaptic neurons that respond to stimulation by releasing chemicals that travel back to the presynaptic terminal where they inhibit further release. So this is the process by which basically the neurons say, hey, there's enough neurotransmitter, stop releasing it. And that's done both from the autoreceptors in the presynaptic neuron and postsynaptic neurons that release chemicals that then signal to the presynaptic terminal to inhibit further release. So to summarize this whole process, an action potential is transmitted down the axon to the presynaptic terminal. When the action potential reaches the presynaptic terminal, it opens ion channels for calcium to enter the neuron. Calcium causes synaptic vesicles to fuse with the terminal membrane, open and release neurotransmitter molecules. Transmitter molecules bind to postsynaptic receptors, causing ion channels to open or close, and ions to move into or out of the postsynaptic cell. Ion flow may increase an excitatory, may produce an excitatory or inhibitory postsynaptic potential. Enzymes break down some neurotransmitters in the synaptic cleft. Some molecules are taken back into the neuron and repackaged. And of course, some may also bind to presynaptic receptors. So I want to spend the next few minutes talking about different neurotransmitters and neurotransmitter systems. So each neurotransmitter binds to several receptor subtypes. So we talk about receptor specificity. So as we start talking about dopamine and serotonin, there's not just one type of dopamine or dopamine receptor or serotonin receptor. There are many different kinds, and they will have different properties that will become important as we move through the semester. Here's a quick summary of the different kinds of neurotransmitters and their families. We have acetylcholine, which is a quaternary amine. We have the monoamines, which include the catecholamines and the indolamines. Indolamines are the serotonins, or the 5-HTs. Catecholamines are norepinephrine, epinephrine, and dopamine. Neuropeptides are primarily involved in pain regulation. Peptides too as well, oxytocin, substance P. Oxytocin is of course a hormone peptide that is involved in things like mother-infant bonding and those sorts of events. Peptides, other peptides like vasopressin, vasopressin and hypothalamic releasing hormones we'll talk more about later on. And then finally there are also gases like nitric oxide and carbon monoxide which can have effects on behavior. I want to spend time talking specifically about each of these different neurotransmitter systems. Acetylcholine is the first of the neurotransmitters to be recognized because of its important peripheral actions, particularly in cardiac regulation of cardiac events. There are what we call cholinergic receptors, which are either nicotinic or muscarinic. These two different neurotransmitter systems occur in different parts of both the peripheral and central nervous systems and have very different roles to play. Nicotinic receptors tend to be involved in things like memory formation, whereas muscarinic receptors are often involved in the peripheral nervous system. Acetylcholinesterase is the enzyme which breaks down acetylcholine in the synapse. As I mentioned previously, there are what we call ACE inhibitors or acetylcholinesterase inhibitors. These include both irreversible and reversible types. The irreversible are generally toxic. These include pesticides and different kinds of nerve gases. What happens is when you block acetylcholinesterase permanently from functioning, the cells that are responding to acetylcholine, particularly the cardiac muscle cells and peripheral muscle cells, will not stop firing. And so as a result, you get muscle spasms, and eventually you end up having cardiac arrest from too much of these pesticides or nerve gases. That's how they work. They cause cardiac arrest. Reversible ACE inhibitors are thought to be cognitive enhancers and are drugs for treating Alzheimer's disease. Because acetylcholine is involved in memory formation, this is one of the targets for treating Alzheimer's disease because the acetylcholine system seems to be particularly affected by Alzheimer's disease. We talk about this then as the cholinergic system. It's associated with learning and memory. And anticholinergic drugs such as scopolamine can cause amnesia. So you can see acetylcholine has projections throughout the cortex. We'll get in far more detail about how all of this works in these brain areas as we start talking about neural anatomy, but including the hippocampus, which is under the surface of the temporal lobes, cerebellum, which is often involved in motor coordination. We have the brainstem, also particularly involved in the cholinergic system. So the nicotinic receptors tend to control our skeletal muscles, and the muscarinic receptors control our involuntary muscles. So both things like your heart, lungs, most of your interior anatomy are controlled by muscarinic receptors. So the catecholamines are synthesized from tyrosine. These include dopamine, norepinephrine, and epinephrine. We're primarily going to be focused on dopamine and norepinephrine, although certainly epinephrine has some important uses. It's terminated by reuptake and through the metabolism via monoamine oxidase. As I said earlier, monoamine oxidase is a target for some older class antidepressants called monoamine oxidase inhibitors, which have some serious and significant side effects. Many of you have probably seen on the back of almost any over-the-counter drug that says consult your physician before taking an MAOI, and that's what those are called, those MAOIs. So norepinephrine is particularly important for functioning of the sympathetic nervous system. We also have axonal projections that travel down the spinal cord where they exert an analgesic action, which is why when we're under a high-stress fight-or-flight situation, we don't perceive pain in the same way once that fight-or-flight situation is over. So the release of norepinephrine produces an alerting, focusing, orienting response, as well as positive feelings of reward. We'll talk about drugs that affect norepinephrine, such as methamphetamine, and how they can create a paranoid delusional state by causing that huge focused alerting instance. We'll also talk about different attentional networks and how norepinephrine particularly affects one of those attentional networks. The dopamine pathways, there are some important ones we're going to talk about. The mesolimbocortical pathway is particularly involved in what we call the reward pathway, and the mesostriatal pathway is particularly involved in motor coordination. We also have the hypothalamus to pituitary gland, which controls hormone release, the substantia nigra to the basal ganglia, which controls motor coordination, and the ventral tegmental area to cortex and limbic system, which is particularly involved in the reward pathway. So these have become very important as we talk about various systems throughout the semester. Serotonin is synthesized from tryptophan. You can see there are many different 5-HT receptor subtypes. 5-HT is simply a shorter way to refer to serotonin. There are 5-HTs 1 through 7. 5-HT1 has both 5-HT1 and 5-HT2 have different receptor subtypes. So that leaves us with 12 different 5-HT receptor types. These are ionotropic, include 5-HT3. The pathways largely parallel the dopamine pathways. So we have the mesencephalic serotonergic cells that project to the thalamus, the hypothalamus, the basal ganglia, and cortex. So very similar to the dopaminergic pathways, but again, we'll talk about these in some very specific circumstances. Glutamate is our major excitatory neurotransmitter in the brain. It comes from the metabolic pathway, particularly the Krebs cycle, or from glutamine via glutaminase. It binds to several receptor types, including the NMDA receptors, the kainate, and the AMPA receptors. NMDA are mediated by both glutamate and glycine serine. The NMDA receptor, which we're going to spend a lot of time talking about because it has some really important components to it, it's also a very complex receptor subtype. It requires membrane depolarization by kainate or AMPA. The NMDA receptor seems to be particularly involved in memory formation when we talk about what's called long-term potentiation. So it's long-term depolarization by these other receptor types. So again, NMDA is involved particularly in memory formation via this long-term process. A couple of other things to note about glutamate is it may be implicated in epileptic or seizure disorders, and drugs like PCP and ketamine are what we call glutamate antagonists. Antagonists are substances that antagonize or reduce the efficacy of a particular neurotransmitter. Agonists are those that boost the efficacy of a particular neurotransmitter. So the NMDA receptor has some particularly unusual characteristics that are important for us to consider. First of all, it can release both sodium and calcium into the cell and allow potassium out. There are glycine sites, polyamine sites, glutamate sites. There are also a variety of other sites for things like benzodiazepines and GABA. that can inhibit or not inhibit or boost the efficacy of these particular neurotransmitter sites. So it's a very complex molecule site. In addition to glutamate, another amino acid, either glycine or serine, needs to be present for receptor activation. It's sort of like a two-key system. And for activation, they not only need to be stimulated by glutamate, but also have to have sufficient depolarization in order for them to function. So in order for this receptor to function, it has to have a slightly depolarized membrane as well as the presence of glutamate. So drugs like benzodiazepines hyperpolarize the cell membrane and make it less likely that this receptor will actually fire, which is the reason why these drugs oftentimes have negative effects on memory, a situation we'll talk more about later. So the NMDA receptors play a critical role in regulating synaptic plasticity by making connections stronger or weaker between neurons. Another important neurotransmitter is GABA, which is gamma-aminobutyric acid. It's a universally inhibitory transmitter found in high concentrations in the brain and spinal cord. It's made from glutamate under the control of the enzyme glutamic acid decarboxylase, or GAD. It's metabolized by GABA transaminase, or GABA-T. There are two receptor subtypes, GABA-A and GABA-B. It's terminated by reuptake with transporters on the neuron or on a glial cell. So GABA is an inhibitory neurotransmitter. It's implicated in a variety of instances. Benzodiazepines are sedatives, which reduce neuronal firing, so they are actually what we call GABA agonists. They actually boost the efficacy of GABA and therefore make neurotransmission less likely, which is one of the reasons why they're sedating and also anxiolytic, or relieving of anxiety. But they also have negative effects on memory by making memory formation less likely. The peptides are small proteins or chains of amino acid molecules attached in sort of specific orders. They tend to affect the opioid receptors, which include the mu, delta, kappa, the endorphins and the enkephalins, and substance P. They are metabotropic effects that utilize a number of different neurotransmitters. They are often called neuromodulators. Their release requires repeated stimulation. Released peptides trigger other neurons to release the same neuropeptide. They diffuse widely and affect many neurons via their metabotropic receptors, and in fact dendrites can release neuropeptides as well as axons, so these do have large diffuse effects. So some distinctive features of neuropeptides is they are synthesized in the cell body. They are mostly released from dendrites, cell bodies, and the sides of the axon. They're released by repeated depolarization, are very diffuse, and have effects lasting much longer time than other neurotransmitters. Quick review of some of the types of neurotransmitter synthesis. Acetylcholine is manufactured through a couple of metabolites or via our diet. Dopamine, norepinephrine, and epinephrine are synthesized from phenylalanine to tyrosine to dopa into its constituent components. And then serotonin is manufactured from tryptophan to 5-HT into its different serotonins. Our next topic I want to cover is just a brief introduction to psychoactive drugs and talk about how drugs affect these neurotransmitter systems. There are drugs that bind to receptors. We'll talk about opiates and endorphins, stimulants, cannabinoids, and dopaminergic drugs. So there are many hallucinogenic drugs that distort perception. They chemically resemble serotonin in their molecular shape. They stimulate serotonin type 2A receptors, or 5-HT2A, at inappropriate times or for a longer duration than usual. That's what causes their subjective effects. So when we're talking about these, we're often talking about MDMA, which is methylene dioxymethamphetamine, or other drugs like LSD. So they do distort our perceptions of time, perceptions of thirst, also affect things like our temperature regulation. Nicotine stimulates the acetylcholine receptors. And benzodiazepines are GABA agonists and act by binding to the receptor and facilitating the inflow of chloride ions into the neuron, thereby making it hyperpolarized and less likely to fire. Opiate drugs and endorphins attach to specific receptors in the brain. The brain produces certain neuropeptides known as endorphins, which is a contraction of endogenous morphines. Opiate drugs exert their effects by binding to the same receptors as endorphins. Most opiate drugs have to be metabolized into active metabolites. So, for example, morphine is the active drug that most other drugs get converted into, or hydromorphone. So oxycodone and hydrocodone have to be metabolized into hydromorphone, and that is done through a variety of specific enzymes. And we're going to talk more about how those work later on in the semester. Stimulant drugs tend to be drugs like amphetamine, cocaine, and caffeine. They stimulate dopamine synapses by increasing the release of dopamine from the presynaptic terminal. Drugs like methylphenidate or Ritalin blocks the reuptake of dopamine, but in a more gradual and controlled rate. And as you know, Ritalin is often prescribed for people with ADD, which the reason why this particular drug works, as well as Adderall, which is D-amphetamine, is because they actually allow the dopamine system to re-exert control over the frontal lobes. Cannabinoids are the active chemicals in marijuana that bind to anandamine or 2-AG receptors on presynaptic neurons, or GABA. When cannabinoids attach to these receptors, the presynaptic cells stop sending action potentials, and this way the chemicals in marijuana decrease both excitatory and inhibitory messages from many neurons. This is the reason why there are analgesic effects as well as other effects of cannabinoids. One important thing to realize is the reinforcing effect of all drugs, whether they are opiate drugs, cocaine, amphetamines, all of the reinforcing effects are due to its effects on dopamine. So while cocaine and amphetamine affect dopamine directly, opiates affect dopamine indirectly. And so that regulation of dopamine can be altered by a variety of different drugs. Last area to talk about in neurotransmitters is to talk about hormones. These are chemicals secreted by a gland or other cells that are transported to other organs by the blood, where they alter activity. They're produced by the endocrine system or the endocrine glands. These include a variety of glands, including our sexual organs, testes and ovaries, kidneys, pancreas, thyroid, adrenal glands, all sorts of areas, including fat cells, can function as adrenal glands. They're important for triggering long-lasting changes in multiple parts of the body. So when we talk about the major endocrine glands, we're talking about the hypothalamus, pineal gland, the pituitary gland, all contained in the brain, the parathyroid, thyroid, and thymus, all there in the thoracic area, the liver, adrenal glands, kidneys, and pancreas, and then of course we have testes and ovaries, and then the placenta in women during pregnancy all have major roles to play in these processes. So, selective list of different hormones we may talk about throughout the semester and where they come from. The hypothalamus relates a variety of hormones, but usually these are hormones that prohibit or inhibit the release of hormones from the pituitary. From the anterior pituitary, we get thyroid-stimulating, luteinizing, follicle-stimulating, ACTH, prolactin, and growth hormone. From the posterior pituitary, we get oxytocin and vasopressin. These are important in very specific areas. The pineal gland is responsible for releasing melatonin, which affects our sleepiness. The adrenal cortex releases aldosterone and cortisol. The adrenal medulla, epinephrine and norepinephrine. Pancreas, insulin and glucagon. Ovaries, estrogen and progesterone. The testes, testosterone. Kidneys, renin and fat cells release leptin. They are also known to alter estrogen and testosterone, so we'll talk more about that later on. So the pituitary gland and the hypothalamus are really the masters of the endocrine system. Attached to the hypothalamus is the pituitary gland and consists of two specific glands. The anterior pituitary is composed of glandular tissue. So the hypothalamus secretes releasing and inhibitory hormones that control the anterior pituitary. The posterior pituitary is composed of neural tissue. So the hypothalamus produces oxytocin and vasopressin, which the posterior pituitary releases in response to specific neural signals. So very specific, separate roles for both the anterior and posterior pituitary. So as we talk about how these affect behavior, oftentimes we have to understand that these are being released into the bloodstream and then traveling to other parts of the body to have their effect. So the hypothalamus releases the TSH-releasing hormone, the anterior pituitary releases TSH, which then travels to the thyroid gland, causing an excitatory effect that then thyroxine and tridothyronine are released by the thyroid gland and then travel back up to the hypothalamus and inhibit it from releasing TSH, which then causes the pituitary to stop releasing TSH, and then the levels of these hormones reduce, and then that stops traveling to the hypothalamus, and then the hypothalamus starts releasing those again. So the hypothalamus maintains a fairly constant circulating level of hormones through a negative feedback system. This is how the thyroid works. This is how testosterone works. And so these are particularly important for understanding how behavior works. Thank you very much, and we will